Good morning, everyone, and welcome. I'm delighted to have you here today. We're about to start a session on cleaning validation, a critical aspect of our quality assurance processes. The focus of this presentation is on MACO calculations and acceptance criteria. Over the next few slides, we'll explore the key concepts, methodologies, and best practices to ensure our cleaning processes meet the highest standards. Let's get started. Today, I want to talk about the importance of ensuring equipment cleanliness, product safety, and GMP compliance in our operations. First, it's critical that we demonstrate our equipment is truly clean after completing our cleaning procedures. This isn't just about visual inspection. We need to verify that residues are removed effectively. Next, we have to ensure there is no cross-contamination between products. Cross-contamination can compromise product quality and safety, so thorough cleaning and validation are essential to prevent that. We also need to provide scientific evidence that any residues left behind are within safe and acceptable limits. This is key to maintaining trust and compliance with regulations. Speaking of regulations, this process is vital for meeting the requirements of agencies like the FDA, EMA, WHO, and PIC-S. They all emphasize the importance of cleaning validation to ensure safety and quality. Now, let's introduce the concept of cleaning validation itself. It's a systematic, science-based approach that confirms our cleaning processes are effective and consistent. This ensures that we not only meet regulatory standards, but also uphold the highest safety and quality standards across our operations. Today, I want to highlight the key objectives of cleaning validation. First and foremost, preventing contamination in the next product is critical. This ensures that each batch is safe and meets quality standards. Equally important is ensuring patient safety, which is our top priority in pharmaceutical manufacturing. We need to maintain the potency and purity of our products, so no residuals or contaminants compromise their effectiveness. Consistency in cleaning procedures is essential because it helps us achieve reliable results every time, reducing variability. Lastly, compliance with global GMP guidelines is a must. Following these standards not only keeps us compliant, but also supports the overall integrity of our manufacturing process. Collectively, these objectives form the foundation of an effective cleaning validation program that safeguards both our products and our patients. Today, I want to talk about some of the key contaminants we need to consider in pharmaceutical products. First, we have active pharmaceutical ingredients, or APIs, which are the core components responsible for the therapeutic effect. But it's important to remember that APIs can also degrade over time, leading to degradation products that might affect safety or efficacy. Next, we have excipients. These are the inactive substances added to help deliver the drug effectively. While they are generally safe, they can sometimes introduce contaminants or interact in ways that impact the product's stability. Cleaning agents and detergents are also critical to consider because improper cleaning can leave residues that contaminate the product, especially in manufacturing environments. Then there's microbial contamination, which is a major concern, particularly in sterile products. Even small microbial presence can compromise the safety of the medication. Overall, these are some of the main types of contaminants we need to be vigilant about, as they directly influence the quality and safety of our pharmaceutical products. Recognizing and controlling these contaminants is essential in maintaining high standards in pharmaceutical manufacturing. Let's take a closer look at what acceptance criteria should really include. First, they need to be scientifically justified, meaning they are based on solid data and understanding of the product and process. This ensures we're setting realistic and meaningful standards. Next, they should be product-specific, tailored specifically to the particular product we're validating. This helps us address any unique risks or characteristics. They also need to be risk-based, meaning we consider the potential impact of residues or contaminants on patient safety or product quality. This way, our criteria are focused where they matter most. In terms of how we express these criteria, there are a few common units. For instance, We might use milligrams per square centimeter or micrograms per swab area to quantify residues directly. Sometimes, parts per million, or ppm, are used, especially for very low levels of contaminants. And importantly, we also have dose-based limits, like PDE or ADE, which relate to the maximum safe exposure levels. All of these elements form the core of acceptance criteria in cleaning validation, ensuring our process is both effective and scientifically sound. Let's talk about MACO, which stands for Maximum Allowable Carryover. Essentially, it defines the highest amount of residue from a previous product that can be carried over into the next one without posing any risk to patients. This is a critical concept because it helps ensure patient safety by controlling potential contaminants. MACO ensures that there is no therapeutic effect from the residuals, meaning it doesn't alter the intended treatment of the next product. It also guarantees there's no toxicity, so no harmful substances are carried over. Additionally, it prevents allergenicity, which is important for patients with sensitivities, and it maintains the overall quality of the product. So, in summary, MACO is about setting safe limits to prevent any unwanted effects from residuals, helping us maintain high standards of safety and quality throughout the manufacturing process. It's a key part of our overall approach to product integrity and patient safety. Now let's look at some of the key approaches used to calculate MACO, or Maximum Allowable Carrying Opportunity. First, we have the 10 ppm criterion, which is a simple threshold often used to ensure safety. Next, there's the dose-based approach, which involves a health-based limit, typically the PDE or ADA, that helps determine safe exposure levels. Then, we have the LD50, or toxicity-based approach, where we look at the dose that causes death in 50% of test subjects to assess risk. We also consider the visual cleanliness limit, which is important in contexts where visibility or cleanliness impacts safety. The worst-case product approach takes a conservative stance by assuming the most hazardous scenario to ensure safety margins are maintained. All these methods are different, but serve the common purpose of establishing safe limits for exposure. By understanding these approaches, we can better appreciate how we set and regulate safe thresholds in various environments, aligning with our overall goal of protecting health and safety. In this slide, we're focusing on the MACO calculation using the 10 ppm criterion. MACO stands for Maximum Allowable Concentration of Odor, and it's a key concept in ensuring air quality standards are met. When we apply the 10 ppm criterion, we're essentially setting a threshold to determine whether an odor is acceptable or not. This helps us establish safety and comfort levels for both the environment and the public. The calculation involves understanding the concentration of odorants, and then comparing it to this 10 parts per million benchmark. By doing so, we can make informed decisions about necessary control measures. Overall, this method is crucial because it provides a standardized way to evaluate odor impact and protect community health. This slide ties into our broader goal of designing effective, scientifically based odor management strategies. Today, I want to talk about the MACO, or the Maximum Allowable Cumulative Dose, using a dose-based approach that focuses on PDE and ADE. So, what does that mean? Well, PDE stands for Permissible Daily Exposure, and ADE is the Acceptable Daily Exposure. These are critical concepts because they help us set safe limits for drug exposure over time. When we use a dose-based approach, we're essentially looking at how much of a drug a person can safely take daily without risking toxicity or adverse effects. This method is especially useful because it allows us to establish clear, quantifiable thresholds for safe drug use. The key point here is that by focusing on PDE and ADE, we're prioritizing patient safety and ensuring that cumulative exposure stays within safe bounds. This approach helps us prevent adverse drug reactions that could occur if doses accumulate beyond safe levels. Connecting this to our overall theme, understanding and applying the dose-based MACO helps us improve drug safety profiles, optimize dosing regimens, and ultimately protect patients more effectively. So, in summary, using a dose-based approach with PDE and ADE allows us to set precise, safe limits for drug exposure, which is fundamental in our efforts to ensure both efficacy and safety in pharmacotherapy. Let's take a closer look at converting MACO to swab and rinse limits. This step is crucial because it helps us establish clear, standardized thresholds for contamination levels. By doing so, we can ensure consistency across testing procedures and maintain the highest quality standards. When we convert these limits, we're essentially translating the acceptable contamination levels from one measurement method to another, which makes testing more practical and reliable. This process ties directly into our overall goal of ensuring product safety and compliance. It might seem technical, but it's about making sure our methods are aligned and that we're accurately monitoring contamination to protect consumers and meet regulatory requirements. Now, let's take a closer look at acceptance criteria at the ppm level. Essentially, these criteria serve as a clear definition of what success looks like for each project or initiative within the portfolio. They help ensure that everyone involved understands what needs to be achieved and what the expected outcomes are. By setting these standards up front, we reduce ambiguity and align expectations across teams. It's important to remember that well-defined acceptance criteria also facilitate more effective validation and approval processes. As part of our overall project governance, they play a crucial role in maintaining quality and ensuring that each project contributes to our strategic goals. So, in essence, clear acceptance criteria at the ppm level act as a roadmap, guiding us from initiation through to successful completion. Let's take a look at a practical example involving ppm limits. Here, we're focusing on how setting a specific ppm threshold impacts our process. Essentially, the ppm limit defines the maximum allowable concentration of a defect or impurity in a product. By establishing this limit, we can better control quality and ensure consistency. It's important to understand that the ppm limit isn't just a number. It reflects our quality standards and customer expectations. For instance, if we set a lower ppm limit, we're aiming for higher quality, but that might also mean more rigorous testing or potential rework. Conversely, a higher ppm limit could reduce costs but might risk customer satisfaction. Connecting this to the overall theme, managing ppm limits is a key part of process control and continuous improvement. It helps us balance quality, cost, and efficiency, ensuring we meet our goals without overextending resources. So, understanding and applying the right ppm limit is crucial in maintaining a robust and reliable process. Let's talk about visual inspection. This method involves checking the equipment visually for any residues, and when we see no visible residues, we refer to that as NVR, or no visible residues. It's a simple yet effective way to quickly assess cleanliness. Keep in mind, visual inspection is complementary to analytical methods, meaning it works alongside more detailed tests to give a complete picture of cleanliness. Moving on to rinse sampling, this technique allows us to sample the entire equipment train by rinsing it thoroughly. It's particularly useful because it captures residues from all parts of the system, including areas that might be hard to access visually. This makes it a valuable method for ensuring the cleanliness of the whole process line. Then we have swab sampling. Here, we're typically swabbing a specific area of about 25 square centimeters. This method is especially helpful for targeting difficult-to-clean areas or spots where residues are more likely to hide. It allows us to focus on critical zones that need closer inspection. Overall, understanding these sampling methods and how they complement each other is key to maintaining high standards of cleanliness and ensuring product safety throughout the process. When we're talking about methods for residue detection, it's crucial that these methods are validated. This means they need to be proven to be reliable and accurate for the specific application. Sensitivity is also key, and it must be below the MACO, or maximum allowable carryover, to ensure safety and compliance. It's equally important that these methods are specific to the product or cleaning agent we're testing. You don't want to get false positives from other substances. Popular analytical techniques include HPLC or UHPLC, which are highly precise for separating compounds. TOC analysis helps us measure organic carbon content, giving insight into organic residues. UV/Vis spectrophotometry is useful for detecting specific compounds based on their light absorption. Conductivity measurements can indicate the presence of ionic residues, and gravimetric analysis involves weighing residues directly for quantification. All these methods are part of our toolbox for ensuring that residues are within safe limits, maintaining product quality and safety. Connecting this to the overall theme, it's about choosing the right, validated method to accurately monitor and control residues in our processes. Let's now look at the key elements involved in our cleaning validation process. First, we'll consider the equipment description. It's important to understand the specific equipment being validated, including its design and materials, as these factors can influence cleaning effectiveness. Next, product selection is crucial. Here, we focus on the worst-case scenario, meaning the most difficult product to clean or the one that leaves the most residues, to ensure our process is robust enough for all products. Moving on to cleaning procedure details, we need to define exactly how cleaning is performed, including the agents used, contact times, temperature, and any rinsing steps, as these all impact the cleaning outcome. Our sampling plan should specify how and where we collect samples, ensuring we accurately assess cleanliness across different equipment areas. Analytical methods are equally important. They must be sensitive enough to detect residues at acceptable levels, whether in ppm, micrograms per square centimeter, or micrograms per swab, depending on the parameter. Acceptance criteria set the threshold for cleanliness. These are the maximum allowable residue levels, and they need to be clearly defined to determine if cleaning is successful. Regarding validation runs, we typically perform multiple iterations, say at least 3, to demonstrate consistency and reproducibility of the cleaning process. Revalidation requirements come into play whenever there are significant changes to equipment, products, or cleaning procedures, ensuring ongoing effectiveness. Finally, we need a comprehensive cleaning validation protocol that documents all these steps, providing a structured plan to follow and ensuring compliance with regulatory standards. All of these components work together to establish a reliable, consistent cleaning process that guarantees product safety and quality. Now, let's review what should be documented in the cleaning validation report. First, we need to include all observed data, which covers every piece of information collected during the validation process. Next, we should document the analytical results, highlighting the outcomes of our testing and measurements. It's also important to note any deviations that occurred and the corresponding CAPA, or corrective and preventive actions taken to address those issues. Additionally, a thorough statistical evaluation is essential to support our conclusions, ensuring the data is sound and reliable. Finally, based on all this information, we can confidently conclude that the equipment is clean and acceptable for use. This comprehensive documentation not only provides transparency, but also demonstrates compliance with our quality standards and regulatory requirements. Now, let's look at some of the key regulatory guidelines that shape our manufacturing and quality standards. First, we have the FDA's 21 CFR Part 211, which sets the current good manufacturing practice requirements for pharmaceuticals in the United States. It covers everything from personnel and facilities to production and quality controls. Then, there's the EU GMP Annex 15, which provides guidelines for validation and qualification processes within the European Union, emphasizing the importance of a risk-based approach. Next, we have PIC SPI 6, which is part of the Pharmaceutical Inspection Cooperation Scheme's guidelines, promoting harmonization and mutual recognition among inspection authorities worldwide. Moving on, the WHO Technical Report Series offers international guidance, especially useful for ensuring quality in global supply chains. Additionally, the EMA's PDE guideline from 2014 addresses the permissible daily exposure levels for impurities, helping us manage risk and ensure patient safety. Overall, these key regulatory guidelines serve as the backbone of our compliance framework, ensuring that we meet international standards, maintain product quality, and ultimately protect patient health. Cleaning validation is a critical step to ensure that our equipment is safe and free of any residual contaminants. MACO plays a key role here by establishing a scientific carryover limit, which helps us determine acceptable levels of residues. These acceptance criteria are often expressed in terms of parts per million, milligrams per square centimeter, or micrograms per swab, depending on what's most appropriate for the situation. You'll notice that the 10 ppm standard and dose or PDE-based methods are widely used across the industry. These approaches help us set clear, measurable limits that align with regulatory expectations. Ultimately, the goal is to ensure GMP compliance, maintain high product quality, and most importantly, safeguard patient safety. So, in summary, thorough cleaning validation with well-defined carryover limits is essential for our overall quality system. It ensures that our products are safe, effective, and meet the strict standards we're committed to upholding. For further support, please contact us. We're here to help if you have any questions or need additional information. Thank you so much for your time and engagement today. Remember, we're committed to assisting you every step of the way, so don't hesitate to reach out.